Okay, hello everybody. We're going to start our AP Bio lecture on the cell unit. We're going to start with the basics. So let's go ahead and you don't have to write these down because this is where we're going. So this is kind of a map of where we're going. We'll talk about membranes, we'll talk about the nucleus, we'll talk about the cytoplasm, then we'll break down each individual organelle. Okay, so let's start with most of the time this is sort of like a pretty good advanced cartoon version of a cell. Just understand that a lot of this stuff is fake colorized to kind of show you where it is. Like you wouldn't see these real colors in a cell. And don't forget that a lot of the microscope pictures that you guys look at are stained. So cell colors aren't quite this dramatic. So here's a typical plant cell. Big difference being it has these cell walls. And a couple things that we don't see in plant cells that we do see in animal cells are listed down here. Plant cells don't have lysosomes to break things down. They don't have centrioles, and they don't have flagella because they don't really move around. But you do see some plant sperm that have them. But a typical diploid cell doesn't have them. Of course, they have chloroplast, and animal cells do not. Okay, let's start with membranes. Okay, so if you're going to do cellular chemical reactions, we want to keep the things inside of the cell. We want to allow all these reactions on the inside to happen in a controlled space. And so we have membranes that separate the whole cell from the environment, but we also have compartments inside cells that we're going to call organelles that specialized reactions will happen in. So you can think about this as like your chemistry lab. You can think of these as like individual test tubes or beakers or places where reactions are happening in controlled areas. Okay? The other thing it does is makes a boundary for regulating what comes in and what comes out. We don't want every little thing that's out in the environment. We don't want every little thing that's out in the environment to come in. So some things we want to sort of block and not let them enter the cell. But there are some things we really want to come in. So we might want nutrients and water to come in. So we'll say, yes, you can come in. Over here, no, you can't come in. So it's sort of a regulator for what comes in and what comes out. Okay, so let's talk a little bit about the cytoplasm or cytosol. And we'll do more on the membrane, so that's just a basic overview for now. So your cytoplasm or cytosol, either one, that's what's between the cell membrane and the nucleus. So if you set up a cell, all this sort of space right here where I'm drawing these dots, that's a very fluid, very watery substance that just kind of allows these organelles to sort of float. And it also gives like a pathway for things to move. Liquids are really, good for moving chemicals around. So if you're going to move things around, it's sort of like a pond. Okay, you can float things on one side or the other. I was going to say a river, but rivers kind of have an in and out. This does not. So there's a gel type and a sol type. So those are based on their viscosity. All right, let's just talk about what an organelle is before we go into the individual ones. So organelle means like little organ, okay? And it's a compartment in a cell inside of a membrane. So that's sort of like a big thing to add here is most of them are in membranes. Now, not all of them are, like cytoskeletons and centrioles, but a lot of them are in membranes, okay? Because that's where we get compartment. So what does this do? This keeps the cell organized and allows certain functions to happen in certain containers. All right, let's look at some functions here. Organelles are a way, like I've said, I just said this already. Sometimes I get ahead of myself. They're a way to separate chemical reactions. There's some chemical reactions you don't want to have out in the cytosol. You want to have those in controlled, concentrated areas where you can, it's like mixing something up. You don't go into the kitchen and say, let's just add one of everything into what we're going to make. You only let certain things into your bowl or whatever you're making your food in, because you want to just bring together just the ingredients you need. And when you bring just the ingredients you need together, chemical reactions tend to take place better in areas where they're concentrated and they don't have other chemicals to interfere. Okay? It also keeps the enzymes that do all this work separated. So, when you compare, let's just stop for a second. This is a eukaryotic cell. It has different compartments for things to happen in, where this might be a prokaryotic cell. And the prokaryotic cell, everything has to happen in this general space. So the eukaryotes have a much more advanced cellular chemistry set, okay? They are able to do things much, much more efficiently. A prokaryote might absorb some sort of nutrient, and it just kind of has to happen in that general space, where over in the eukaryote, we might concentrate that into a specialized space, okay, called an organelle. All right. So what do you have to do when we get done with this unit? You got to be able to tell me what the major organelles are. And you just did your project where you looked at an individual organelle, but you got to sort of become an expert on all of them, not just the one you did. Talk to me about their structure, okay, their shape, their compartments, and what happens in those. Tell me what their job is, okay? So structure and function is a theme throughout the entire class, okay? So these words will come up over and over again.